Hello, everyone. Thank you. Thank you, Tatiana, for the introduction. And wasting no time, I move on the presentation. In non-scientific literature, but also in taxonomic or phylogenetic papers, we can find many terms regarding structures on the surface of insects' body. These are terms such as bristles, hetae, setae, spines, spinal tubercles, pilosity, pubescence, and hairs. But of course, the term hairs refers only to the structures that grow from fossils found in the dermis of mammals, which constitute an autapomorphy for this group of animals. Most of these structures are sensory organs, sensilla. And here we face the first problem: how to distinguish sensilla from other cuticular structures. Theoretically, it is simple. Sensillum is characterized by having at least one neuron that is absent in non-sensory organs. Unfortunately, checking this requires sophisticated methods and advanced light microscopy or electron microscopy. Its dissemination took place in the 70s of 20th century, and one of the first studies focused on Miridae trichobothria. The method that facilitated and popularized the research of sensory organs is scanning electron microscopy. Okay. A suitable scanning microscope allows determining the function of individual sensilla based on their morphological features. The advantage of this method is that the preparation of material for testing in CIM is relatively simple. Functionally, there are three types of sensilla: Mechanoreceptors, Chemoreceptors, and Thermo and Hygroreceptors. The same sensillum is usually responsible for temperature and humidity perception. The morphological division, on the other hand, is much more complicated. Researchers use various, usually incompatible classification. For example, this sensillum, we can find a whole of terms in the literature, such as sensilla basiconica, multiporous placodea sensilla, multiporous olfactory sensilla, or sensilla tychoidea, and others. But it is not everything. Many subtypes are distinguished, indicated by letters, numbers, or shape names. The terminology of sensilla definitely needs to be revised. In addition, some taxa have sensilla with very characteristic shapes not found in other insects, e.g. in waterbugs, leaf-shaped, club-shaped, brush-shaped sensilla, and other are known. But the information provided in this presentation will be only about terrestrial true bug. Most of the large sensilla on the insect's body are mechanoreceptors. These receptors can perceive mechanical stimuli such as touch, sound, vibration of air, water, and ground, as well as tension of bending the cuticle. So they are responsible for the sense of touch, hearing, and proprioception. Cuticular mechanoreceptors can be divided into two types: protruding hair-like sensilla with flexible socket, and campaniform sensillum characterized by presence of dome instead of appendage. Flexible sockets allow for deflecting sensilla, which stimulates the dendrite types. Hair-like mechanoreceptors can be modified in various ways, e.g. flattened or shortened. And a quite spectacular example of mechanoreceptor modifications are spinous tubercles occurring in the inner surface of the Metaphomora in Femurocoris, New Caledonian representative of Miridae. Perhaps the most recognizable type of sensiloop in Heteroptera is Trichobothrium. It's trich situated in a cup-like depression in the cuticle, bothrium, or a small dome surrounds of its base. Trich is fine and long, everything almost along their entire length, flexible and tapered apically. Their surface is characterized by parallel grooves. Bothrium is frequently surrounded by trichome, an area of densely arranged microtricha. Despite many characteristic features, Trichobothria shows considerable diversity. For example, on the Miridae femora, we can find those with a typical structure as well as reduced ones. The number and arrangement of Trichobothria are highly conservative, and therefore they are important in the diagnostic of family group taxa and phylogenetic studies based on morphology. The diversity of Trichobothria positions on the body in various taxa of Heteroptera could indicate that these sensilla have different origins. The Trichobothria are located on the head, antennae, as in the case of Reduvidae, scutellum, labium, and abdomen, for example, in Legaoidea. And in the Miridae, 3 to 8, rarely more, the Trichobothria are located on the lateral and ventral surfaces of the meso and meta femora. This is an important diagnostic feature of this family. In 2030, Liu Sigvald and Cheho observed a pair cephalic Trichobothria in the immature stages as well as in the imago of Fulvius carioni. In the conducted studies, I have demonstrated that the presence of ventral cephalic Trichobothria is a characteristic feature of plant bugs. A pair of ventral cephalic Trichobothria was observed in all examined species. Each Trichobothrium of this pair is located laterally to the first article of rostrum on the gula between the buccula and the antennal tubercle. Moreover, a dorsal cephalic Trichobothria was observed for the first time. They are located above the antennal tubercle towards the center of the frons. Chemoreceptors are responsible for the senses of taste and smell in insects. The olfactory receptors detect gaseous chemicals, and the taste receptors detect substances dissolved in water, even if in dry surfaces. To detect chemical substances in the air, the sensillum must have pores on its surface through which molecules can enter to interior, where the dendritic ends are located. The olfactory receptors are found on the antennae and the labium. Taste sensing contact chemoreceptors usually have only one pore of the apex of the appendage, which is usually hair or peg-shaped. Taste receptors are located on the tip of labium. The olfactory receptors on antennae can take various shapes. There are often trichoid, but also hetica, basiconica, or celoconica sensilla. Unlike mechanoreceptors, the chemoreceptors usually adhere to surface of the antennae. As I mentioned, these sensilla have numerous pores, which are visible only with high magnification. The pores range from 10 to 25 nanometers in diameter, and their distribution varies depending on the type of sensory organ. Sometimes the same sensillum has a dual function, being both mechano and chemoreceptors. I will present chemosensory organs on the tip of the labium on example of neighbors. Nevertheless, preliminary research results on Miridae indicate that they have very similar chemoreceptors organizations on the labium. In addition to the mechanoreceptors, numerous marked yellow and a pair of orange, there are 2 and sometimes 3 types of chemoreceptors in the labium. Marked with a light blue color, PEC sensilla with a terminal pore are responsible for the sense of taste. Elongated placoid sensilla with wall pores, red, and oval plate sensilla with terminal pore, brown, are olfactory organs. In addition, between the gustatory organs, there are 4 sensilla that function as Thermo and Hygroreception, marked in dark blue. It is noteworthy that the number of chemoreceptors and Thermo and Hygroreceptors appears to be a conservative feature in all studied Navidae, Miridae, as well as individual species from several other families have the same numerical scheme of this sensilla. Apart from the Thermo and Hygroreceptors of the labium, which I have just mentioned, the sensilla responsible for these senses are also located on the antennae. The most common are sensilla coeloconica, a non-pore PEC inserted into a pit. This type of sensillum is found in small numbers, and the mechanism of action of hygroreceptors is poorly understood. Probably changes in air humidity affect the water content in the sensillum cuticle, causing its deformation. Of course, not all structures on the body of true bugs are sensory organs. The hair-like appendages are assigned a covering and protective function. However, the role of many structures is unknown. Example includes thread-like structures described by Ibrahim and colleagues, or the cuticular circular formations we observe on both antennae and the legs of Miridae. There is also a secretory pore system on the antennae, often associated with the basis of the olfactory sensilla. And this one is also interesting, whatever it is. I have seen a similar structure in mites. It was referred to as trichobothria, but I have any idea what is this. And an interesting issue is teratological cases of the antennae. Antennal anomalies can originate in the nymphal instars through injuries to the antennae and their subsequent regeneration during the neck instars. Antennal anomalies in Pentatomomorpha have been observed much more frequently than in other infraorders. For example, in a study conducted on Aradidae, we found malformation of antennae in 2.9 of the cases. However, within the new species of Meschia, family Meschidae and superfamily Legaoidea, as many as 8 out of 33 specimens, for 24%, were characterized by antennae teratology. Oligomere, otherwise called symphasomere, which depends on reducing the number of segments that results from injury and the anomaly that has most often been reported. Interestingly, although the number of segments is smaller, they are larger than normal and the characteristic set of sensory organs is reproduced. This phenomenon is known as compensatory regeneration. Maintenance of sensory functions of the teratological antennae may be helpful in better understanding the functioning of the sensory organs. And conclusions. Most of the microstructures on the surface of true bug's body are sensory organs. Diversity of sensilla is certainly greater than previously assumed. There are cuticular structures whose function is completely unknown. And the determining the homology of sensory structures and their phylogenetic and taxonomic significance requires further detailed ultramorphological studies that should include a wide range of taxa. I want to extend my sincere thanks to everyone who contributed to the results presented today, especially thanks to Adrian, who spent hundreds of hours with me working on CIM, and Professor Jolanta Brożek, who introduced me to the subject of sensory organs a few years ago. And by the way, if you know any species with unusual antennae morphology or with interesting structures, I would be grateful for the information. Thank you for attention.
Thank you very much.
Wonderful.
Thank you very much for giving me this time, first of all. So yes, it was mentioned before, I'm gonna go an overview on what is the current situation of the family Pentatomidae with the recent results that have been going on. So first of all, present like the group, why it's important. We know like pentatomids are important plant pests with one of the most important plant pests currently with a worldwide distribution, Alimorphalis, which is basically eating all the crops around the world. But also pentatomids are important because they can act as biological control agents. There is a small group that are that feed on other invertebrates, mainly holometabolous insects, which is the asopines. And then we cannot forget the fact that many species are consumed locally as food, providing a good source of proteins to the diet. So first of all, previous to the analysis, the family was already recovered as monophyletic using a small data set of, or a small number of species, and using both morphological and molecular markers. The family is characterized by having a set of synapomorphies like the Gonapophysae and the first Ramilus, the presence of triangulin, the Gonapophysis 9 reduced fused to the Gonacoccyx 9 with the second Ramilus, and finally the spermathecal duct is dilated and its distal orifice is provided with a sclerotized rod within its side. So with the information we had previously, however, the internal relationships of the Pentatomidae were not solved. Some of the subfamilies appear to be polyphyletic or non-monophyletic in this total evidence analysis. However, nothing could be extracted from that, as the amount of taxa included in the analysis was pretty low in terms of the family Pentatomidae, as the aims of this study was the relationships within the superfamily. Also other results obtained from other molecular markers, in this case obtained from whole mitochondrial genomes, obtained pretty similar results to ones obtained in 2008. However, also with a very small amount of taxa, which therefore could not lead us to any information. As we can see, the results were partial, however, highly congruent with the previous ones obtained. Some of the groups within the Pentatomidae, however, have we have obtained, or groups have obtained phylogenies of them. And as in the case of the Antisine and the Discocephaline, they both were recovered as monophyletic, and the internal relationships were recovered too. The same case happens to some of the groups within the subfamily Pentatomidae, which is currently a dump group in the Pentatomids, however, the nominal subfamily. And this is the case of some of the Asian Aliini, the Euchistus group, or more recently, the Chlorocorini. However, in all the previous results obtained, suggested that Theratochorids were placed at the basal lineage, as some of these orders already mentioned. However, after from the results of the phylogeny obtained, Theratochorids were excluded from the family Pentatomidae, which was already hypothesized by several authors. And as we can see here in the amplified part of the phylogeny, we can see that clearly Theratochorids fell totally apart from the Pentatomids, and their status was renewed as a family by itself. And this is after examining the Theratochorids, this makes some morphological sense, as they have, in one case, absence of triangulin. The second valvifer has a distinct medium fusion line, and the male phaloteca is relatively flexible, which is not the case in Pentatomids. However, what made some others think that Theratochorids might fall into the Pentatomids is the fact that the spermatethal duct is dilated and it has some kind of sclerotized rod within it, which in this case would be from a different origin. So after the results obtained in the phylogeny, including so far the most number of taxa, we could decide that the family is composed by 9 subfamilies, or 9 classical subfamilies until what we consider subfamilies until date. The subfamily Cervaninae, which was questioned if it belonged to the family Floidae or not by previous authors, clearly was separated from the Floidae and fell into a early diverging lineage of the Pentatomids. Also, if we compare the internal female genitalia of these two groups, we can see that they are clearly different. And in the case of the Cervaninae, they follow this general Pentatomidae structure organization. Additionally, in the analysis, we found that Rhynchocorines, which is currently a tribe within the Pentatomidae, always appeared as one of these early diverging lineages within the Pentatomidae. And this, in this case, this was obtained in previous results also, and also some unpublished results based only on morphology that I could extract from my dissertation. Additionally, some curious cases, the neotropical groups Chlorocorini currently in the Pentatomidae and the subfamilies Discocephaline and Edesine, all from the neotropical region. They kind of form a group which we really do not know yet the relationships among them, but they are always they always appear as early diverging in the tree of the Pentatomidae. And this was also congruent with the previous results obtained from research that came out previous to the general phylogeny. So after these groups were more or less stable and the tree resulted from that gave us some clearly insight, we happen to have a big bunch of stuff that we really do not know what's going on there. However, besides that, we can more or less get some things clear. However, the relationships of these groups are still not well supported. Therefore, some groups could be recovered as monophyletic. Some groups were clearly not monophyletic in the analysis. And then, as some curious cases, some of the subfamilies that are currently recognized were recovered as sister lineages of Pentatomidae tribes, which we will go on further in the presentation. So in first case, we can say that philocephalines and asopines are monophyletic with the amount of taxa that we included in the analysis. But on the other side, podopines were not recovered as monophyletic. These results were clearly were already stated by previous results, where podopines did not cluster together in a monophyletic group. However, in this case, by including more data, we could clearly see this differentiation. However, we still have all these Pentatomidae sorted out together and without any kind of sense or logic. But unluckily, with the tree obtained, we had no support to establish any information or any classification based on that. We are sure that Pentatomidae is not monophyletic, as clearly the neotropical chlorocorines, the rhynchocorines, and then some tropical bugs that were placed in the Pentatomidae clearly fall out of the rest of this group. On the other hand, Carpocorini, which is one of the biggest groups within the Pentatomidae, is composed by multiple lineages. We can find two main clusters, which one corresponds to the old world and the other one corresponds to the new world. On the other hand, the old world Carpocorini was found to be paraphyletic because the current tribes Agonostelini, and Nailini were clustered within this tribe, which these tribes probably will need to be synonymized under the Carpocorini sensu stricto, while the New World Carpocorini probably needs to be redefined as a tribe by itself. On the other hand, we checked with the phylogeny that some of the characters that are currently used to determine tribes are actually not that stable or should be taken into account in a broader sense. This is the case of the tribe Nephagini, which this tribe is characterized by the specimens or the species being green when alive and green when dead. This might sound a bit absurd, but according to the results we obtained on the tree, all green species in life that remain green in death clustered together in a monophyletic lineage. However, we had some additional species or groups of species that clustered together in this case. This is a case of tribes Miridae, which are very adapted to their environment as they are leaf or Poathia feeders. Some Antestini and some Pentatomini and some other place genera that fell inside this cluster. On the other hand, Menidini was found to be the sister lineage of the Asopines, and that obviously made at least me think, if Asopine is currently one of the subfamilies within Pentatomidae, it cannot be the sister lineage of Menidini, which is currently a tribe within the Pentatomidae. And after examining specimens and checking the different parts, as Asopines had a very clear synapomorphism due to their rostrum specifically designed for feeding on other arthropods. However, checking on the genitalia, we could find that Menidini also possess some of the structures that are not unique to Asopines, but that all Asopines possess, that are the pseudoclaspers present on the male genital structure. This has led to find papers that tend to use the Asopines as outgroup of the pentatomides or of other groups, leading to wrong phylogenetic interpretations, and therefore leading also to possible wrong classifications within this new classification that we are trying to work with the Pentatomidae. So until date, what we have found extracting the morphology from the tree, even though there's not total evidence tree presented yet, we have seen that ventral thoracic structures had not been considered as important in characterizing groups within Pentatomidae, as most of these basal groups, or early divergent groups, better said, all possess some kind of ventral thoracic structure modification within most of the species. Additionally, genetically characters that have been considered, but only in terms of smaller groups, were not interpreted in a whole of the Pentatomidae, and therefore they should be interpreted in the future in order to clear which groups share this set of characters, and showing that some characters should be reinterpreted in a way different to what we are doing until now, like, for example, the rostrum in Nasopine. So for what we have now, the conception of what we have now of Pentatomidae, which we have clear subfamilies and clear tribes, maybe we don't have that much clear information, and we should start thinking that maybe what we thought that they were subfamilies, they might be tribes. What we had as tribes might be subfamilies, and what we thought they were tribes, they're just a bunch of species that group together that have some kind of morphological similarity. So I'm sorry not to have provided you with a whole new classification of the pentatomids, if that was what you were expecting. And luckily, we have still much more analysis to do using combined data sets, also with new approaches, UCs, morphology, in order to produce new phylogenetic studies. Obviously, new taxonomic revisions are needed because some of the groups are complete chaos, and therefore new species, genome revisions, and descriptions. And then all these will hopefully sooner than expected will happen to deliver us some new systematic accounts. But for all these, we will need huge international cooperation, as family Pentatomidae is one of the largest groups, and still with the previous results obtained, one of the major issues we had was the taxon bias, or the lack of samples from certain groups and certain areas. So still, as you have seen with the presentation, there is still loads of work to do. And I put this picture here of Artur Taszakowski, which some of you might have seen, I hope so, because we really do not know where does this fella fall, and it's currently one of the subfamilies within the Pentatomids. And if you happen to have any specimen that we could extract some DNA from it, that would be extremely helpful for a future analysis, and also to know where this fellow falls in the tree. So thank you very much for your attention. I hope that I created even more questions than before with this presentation. That was a bit of the aim, to create some mental instability in how the internal organization of the Pentatomids is. And please, I'll answer any question. And also, I would like to say, just to end up the presentation, remember that the next DHS meeting is occurring in Barcelona from the 4th to the 8th of July, hosted by Dr. Marta Goula and me. We have everything set up, everything's in place. We are just finishing and ultimating the last details. And also remember, all of you, that there's 2 workshops going on during the Congress. One is the workshop on Guerramorpha, led by Dr. Felipe Moreira and Dr. Lana Cheng. And then there's second workshop on Pentatomidae, which I'm trying to lead together with some of the colleagues, in order to basically try to understand what is going on with the Pentatomidae, and more specifically with the Pentatomidae. And thank you very much for your attention.